issue with audio and video. I hope you can hear me now and I'm visible, screen is also visible. So can you please give me thumbs up on the chat box if you can hear me, or you can see the slides for the audio video. Kindly give a thumbs up. I really appreciate your patience. Thank you, Robin. Thank you, Vandan, Hank, Robin Khatri, Deepa, Harun. I really appreciate your patience. So I'll start from the beginning, right? Because many of you never heard it. So we will do it. In this session, I'll welcome you all on the behalf of DBMCI, myself Dr. Varsha Gupta, and I'll be going over the biochemistry recall questions from the NICT, from the NEET PG and from the AIMS also. So because we know the recall questions are very, very important, there are very few questions which they ask over and over. So those could be very easily scoring marks for you guys. Okay, so first I'll go over NEET PG 2021, right? So first question in this is the corneal transparency. Okay, corneal transparency is decided by, there are 4 options: chondroitin sulfate, hyaluronic acid, keratin sulfate, heparin sulfate. These are the 4 choices. I will go over all the 4 choices, explain a little bit about them, right, and then also the correct choice of all of them. Okay, so first, chondroitin sulfate. Okay, first, chondroitin sulfate is what? It is an unbranched polysaccharide of the variable length. It is made up of the glucuronic acid and the N-acetyl D-galactosamine, and it is involved, deficiency of this is involved in osteoarthritis and the coronary artery disease, and its supplement is given with glucosamine. Right, so glucosamine supplements are with the chondroitin sulfate. Remember this. This can be another question in the exam, right? Second choice, hyaluronic acid, right? It is anionic and non-sulfated GAG. Where it is found? It is found in the connective, epithelial and the neural tissues, which is the major component of the extracellular matrix, right? And it contributes significantly to what? To cell proliferation and the migration, cell migration, and it is a natural substance which is found in the eyes and the joints. So it is says the eyes, so you might think it is related to the corneal transparency also, but it major function of it is the cushioning and the lubricants, right? That is what the major function of hyaluronic acid. Third is the keratin sulfate, which is the correct answer for this choice. How you are going to remember it? First, let me tell you about this. Remember this, K is keratin, K corneal, right? So corneal transparency is related to keratin. You may ask, what about this K? This is co. Okay, so we are not picking the co, but K is with the K, that is keratin sulfate, right? It regulates what? The organization of the collagen fibrils and maintains the corneal hydration also. You can very well imagine if there is a deficiency of the keratin sulfate, that will lead to what? Let me change to the highlighter. That is going to lead to the decreased hydration and also corneal opacification, right? And the corneal wound repair will also be negatively affected. They may trick you in exam also by giving the chondroitin sulfate and the dermatan sulfate. As I have told you, the first choice, that is chondroitin sulfate, is not the choice, correct choice for this. Dermatan sulfate is what? It is kind of an isomer of the chondroitin, right? It is participating in the collagen. It does affects the collagen and it also does affects the corneal transparency. But the major function, like keratin sulfate, is the majorly responsible for the corneal transparency. So pick keratin, corneal, don't pick co. Okay. The fourth option, heparin sulfate, they may give you the heparin sulfate, they may give you the heparin also, and that is, we know, is a polysaccharides, right? This is one of the proteoglycan which regulates various biological activities, majorly where we see it in angiogenesis, blood coagulation, right? And it is related to GRB, the gran enzyme B. I'm not going in detail, just know this is gran enzyme B related. And if it is related to what, the SARS-CoV-2 infection also, right? Particularly when it has to bind to the ACE receptors, there that is the point where the heparin sulfate is playing a major role. Okay, so these are the four choices, and the corneal transparency is decided by choice C. That is the correct choice. That is the keratin sulfate. There is this next question in which a 5-year-old male patient presented with anemia, bone pain, hepatosplenomegaly, right? Biopsy is shown here via the following image. Which is the enzyme defect involved in this case? You can keep typing the answers of this question also, right? I'll check you. Robin Khatri, yes. Keratin C. So this question is what? This question, this image, from this image question, what do we see? We see these 3 or 4 big wrinkled tissue paper images, and we know whenever there is this wrinkled tissue paper kind of appearance, that is related to the Gaucher disease, and Gaucher disease is because of GLU. GLU is glucocerebrosidase enzyme, right? So which enzyme is deficient? Glucocerebrosidase enzyme is deficient, and this disease is autosomal recessive and lysosomal storage disorder, right? So our correct choice is A. I'll discuss the second, third and fourth choice also. Hexosaminidase A would be the choice that will lead to what? The Tay-Sachs disorder. And what do we see in We see majorly patient have lots of neurodegenerative disorders, right? And that is because of what? Hexosagangliosa, right? Hexosa is gangliosa. That is because of the accumulation of ganglioside in the brain and the nerve cells, which is going to give rise to any deposition in brain and nerve cells will give you the neurodegenerative disorders. Okay. Now the third choice, iduronidase sulfatase is a lysosomal storage disorders, also falls under the category of mucopolysaccharidosis, right? What is there? There is an excessive excretion of what? The product in the urine, right? And then there is a progressive cell, tissue and the organ dysfunction in this case. Glucose 6-phosphatase is most commonly, one of the most commonly asked enzyme in the exam in one way or another way, right? This is what? That is related to glycogen storage disorder type 1. And type 1 also has 2 subtypes, type 1A and type 1B. Right, and what you will see in the question, in the clinical presentation of the patient, there is poor tolerance to what? Poor tolerance to the fasting. There will be growth retardation and hepatosplenomegaly, right? And what may else they give you? They may give you the accumulation of the glycogen and fat in the liver. Now the next question is question number 3. In an experiment conducted by a student on electron transport chain, when pyruvate and one inhibitor is added, right? Pyruvate and one inhibitor is added, it resulted in the inhibition of what? Oxidative phosphorylation. Which inhibitor was most likely used by the student? Oligomycin, 2,4-dinitrophenol, that is another very important question. Antimycin and rotenone, right? Rotenone is a fish poison. So which one is going to inhibit the oxidative phosphorylation, right? And if we jump back to the class notes, what we have studied in the class? About electron transport chain, it is composed of 5 complexes, complex 1 and 3, right? Complex 1 and 3. Inhibitor of complex 1 is rotenone, right? Inhibitor of complex 3 is antimycin, right? And usually we don't talk about any usually we don't get the inhibitors for the second complex, that is complex 2. What is important about complex 2 is you have the energy, the protons in two forms, NADH and FADH2. The complex 1 is receiving the protons and electrons from NADH. The complex 2 receives the FADH2. So that is why, what do we know? FADH2 is equivalent to 1.5 ATP because the first complex is blocked, and there how many protons goes outside in the inner mitochondrial membrane space? 4 protons. So 4 protons less are going, right? So there will be one less ATP produced towards the end via the ATP synthase, that is complex 5 of the electron transport chain, right? Complex 3 and 4 inhibitors are antimycin A and the cyanide, and this I have given you in the class also, that what is the rotenone inhibits what? If you can remember, rotenone inhibits complex 1, it is a red poison and an insecticide, right? Antimycin inhibits, blocks the complex 3. Cyanide inhibits the terminal transfer, electron transfer. Terminal electron transfer is where? In the complex 4. They may ask you in this way also. Terminal electron transfer, who is responsible? Complex 4 is responsible. And who is the inhibitor? Cyanide is the inhibitor. Carbon monoxide inhibits the cytochrome oxidase, right? What we see always, the cytochrome oxidase, that is inhibited by all the gases such as carbon monoxide, hydrogen sulfide, all the gases, they inhibit the cytochrome oxidase. Now coming back to our presentation, right, the question, right? The question is, which inhibitor was most likely used by the student? Student was using oligomycin with the substrate because it is inhibiting what? ATP synthase. Do you remember ATP synthase? This is the complex here. You have the ATP synthase right here. This is the ATP synthase. It has F1 up there. F1, F0. And oligomycin inhibits what? F0 component. I am unable to highlight it over here, so my apologies for that, but down the screen you can see there is F0, that is part of the ATP synthase. So oligomycin is inhibiting F0, right? More specifically, if you have to answer the question, it blocks proton channel, it blocks the FO subunit, right? Good evening, Health Shine Plus in life, right? So there is no ATP. There is no ATP. Why there is no ATP? Because ATP synthase is helping to bring the proton in. That is, the proton things are built up in between the inner mitochondrial membrane. If that proton is coming via the ATP synthase, only then ATP will be formed. If that channel is blocked, the protons are not coming in, so there will be no ATP synthesis. So that is how the oxidative phosphorylation is being inhibited, right? Right, Ganesh? Yes, F0 and F1, they are rotated, right? It is like a rotor kind of a mechanism that is helping us in the synthesis of the ATP. Since F0 is blocked, the beta subunit of it is blocked, so there is no rotation and there is no ATP generation. Very good, Ganesh Roy. Question number 4. A patient complains of knee pain. Routine investigations are unremarkable and still the patient is unsatisfied. Urine turns black on standing. What is the enzyme involved? So knee pain, urine turns black on standing. What we are expecting here? We are expecting what? A, B, C, D. Which enzyme is responsible? Homogentisate dehydrogenase. This is dehydrogenase. This is where they trick you. we always talk about what dioxygenase or oxygenase or homooxygenase. That is not a dehydrogenase. Yes, homogentisate half portion is right, but this is not the correct choice because dehydrogenase is not the dehydrogenase, it is dioxygenase or oxygenase you will see. So the correct choice is the D. Correct option is the D. Why correct option D? Because this is dihydroxyphenyl acetate. Dioxygenase is the another name of, chemical name of homogentisic acid oxidase. So don't just get lured by this term homogentisate. Look what is behind it. It's not dehydrogenase. It should be dioxygenase, oxygenase, something kind of this. So entire name should be correct. Okay. And this is what we know about the alkaptonuria, right? That is because of deficiency of homogentisate oxidase, right? And that is related to the degradation of your aromatic amino acid phenylalanine, which is an essential amino acid. So from phenylalanine, what we get? We get the tyrosine. That is why tyrosine is a non-essential amino acid, right? Tyrosine gets converted into homogentisic acid eventually, and then it is via the homogentisate oxidase enzyme, we get mellyl acetoacetic acid, right? But if this enzyme is not available, then that will lead to the accumulation of what? That will lead to the accumulation of the homogentisic acid, which will eventually get polymerized in the body, right? And then you will see the deposits in the connective tissues, in the eyes, ears, even in the hands, right? And when the urine is left exposed to the air, then the urine turns black. And when we talk about the symptoms, it is asymptomatic in the childhood. That is asymptomatic in the childhood, and not only just this blacky thing we will see lots of symptoms such as the back pain, arthritic kind of a thing that we see in this, right? And just what is the management? We should be limiting the diet that contains the phenylalanine and the tyrosine, because we cannot degrade and therefore excrete it properly. Yes, Ganesh Roy, in alkaptonuria, tyrosine becomes essential amino acid, right? In the babies, they may give you the sand-like crystals on the diapers, but usually it get unnoticed, right? And this was the mnemonic I gave you. Alka came home, saw black bug and shouted, oh, alkaptonuria, homogentisic acid, black urine, ochronosis, right? And sometimes they may also ask you the question other way route based upon the laboratory diagnosis, right? There is a darkening of the urine with the time. They may not give you the black color, but increased darkening of the urine, positive Bendix test. And remember, this Bendix test is positive because of homogentisate. The ferric chloride test will be positive, right? AgNO3 silver nitrate test will be positive also, and the X-ray will be the parrot beak appearance that you may not see now, but like in the later profs you may see it. Okay. So what did I tell you? This is usually asymptomatic. Usually asymptomatic means it was there since the childhood. The urine was turning black. There was some kind of a crystals in the diaper, but since when that get unnoticed, the features, the physical features are not seen at that point of a time, but eventually in late 30s and early 40s we see the signs and the symptoms, that is erosive arthritis, back pains and stiffness. Is these are the all like two keywords they also ask in the exam, right? And the ochronosis. Ochronosis is what? The black pigmentation of the skin on the tip of the nose, on the ear lobe, elbow, knee, skin, right? Everywhere. And the treatment is nitisinone, or the diet should be restricted. Now this is the question which was there in 2019, 20, 21. Every year they ask this question. So remember, this question is very, very important. Which vitamin deficiency caused this scenario? What do you see this? This is what? This is your Cazal's necklace. Cazal's necklace, whatever way you want to see it. Also this is kind of a rashes, and that is related to what we are talking about, Pellegra, right? Pellegra is related to what? Niacin, right? And niacin is among one of your atypical vitamins, B3, right? And that is synthesized from the tryptophan. So all these things are related. Pellegra is D. All dads are there, right? Dermatitis, dementia, diarrhea, eventually leading to death, right? So if they talk about the niacin and you have the Kezal's necklace, or if you have the Pellegra kind of a symptoms, diarrhea, dementia, right, all these things, then you are going to pick the niacin or the B3 or the tryptophan if the question is straightforward. But sometimes the question is tricky because there are 3 diseases that are related, that revolves around these Pellegra kind of the symptoms. So they may trick you if the question paper is very, very difficult, right? And this is, if you can remember from the class, Pellegra like symptoms revolves around what? It revolves around the tryptophan. If there is a failure to synthesize the niacin from the tryptophan, 60 milligram of the tryp gives you 1 milligram of the niacin, right? Remember this and this. These units must be same. Either they will use the gram or they will use the milligram. We know that, right? So tryp give you this. If the body is unable to synthesize the niacin from the tryptophan, then that will give you hardcore Pellegra symptoms, right? But if there is a failure to absorb the tryptophan, you are getting the tryptophan from the diet, there is a issue, right? Because what is the issue? The transporter is defective. There is this transporter in the kidney and the intestine which is defective, that is going to lead to the Hartnupp's disease. So other than the Pellegra kind of symptoms, you will see other symptoms as well, right? And then there is this carcinoid syndrome also. In carcinoid syndrome, what is happening? The tumor cells are growing at the fast rate. They need lots of the serotonin, right? So all the tryptophan will be rerouted towards the formation of the serotonin, right? There are 2 ways. Tryptophan either forms the serotonin or tryptophan forms the niacin. There are other things it is forming that we have discussed, right? But here majorly remember 2 things, either serotonin or niacin. If it is balanced, both will be balanced, right? In the carcinoid syndrome, this serotonin gets greeded. So all of this will be routed towards the serotonin. So there is no tryp available for the synthesis of the niacin. So hence you will see the Pellegra-like symptoms. Exactly, Wasim. You are right. So next question, question number 6. A 4-year-old boy with developmental delay, recurrent chest infection and worsening bone pain. PTH level is normal, but the calcium and the phosphate levels are low, right? And the patient is also not responsive to the vitamin D supplementation. What kind of a rickets it is? Right, serum phosphate is low, calcium phosphate and calcium is low, not responsive to vitamin D. It is none other than X-linked hypophosphatemic rickets. What is ricket? Ricket is a metabolic bond disorder caused by defect in mineralization because there is not adequate calcium and the phosphate that is available, right? So answer to this is B. Right, and this is the most common form of heritable rickets. This is the correct choice. In this, there is a normal glomerular filtration rate and there is impaired vitamin D3 response. Yes, yes. Right, and it can be inherited in 3 ways actually. If we go in depth, that can be X-linked dominant, autosomal dominant and autosomal recessive. In X-linked dominant, there is FX gene that is mutated, which is going to lead to the increased level of FGF23, right? And there will be decreased what? Renal phosphate absorption. And also this suppresses what? 1-alpha hydroxylase enzyme. That where we have seen this with the vitamin D. If this is inherited in the autosomal dominant fashion, there is a mutation in FGF23, right? So it is degraded. Again, it is going to affect 1-alpha hydroxylase enzyme. if it is inherited in autosomal recessive fashion, the mutation is DMP1 gene, which is going to increase FGFT F23 level. These is not really asked, all these three kind of inheritance, but just to give you few other points, extra edge thing, right? So this is 1-alpha hydroxylase that we see in the synthesis of vitamin D. Vitamin D active form is 1,25-dihydroxycholecalciferol, or you can say calcitriol, right? If this is not working, so here is this one will be get hampered, and that will lead to the rickets in the children and osteomalacia in the adults. Okay, so the nutritional rickets, the choice A is majorly because of the decreased dietary intake. So they should respond to vitamin D. So that is why this is not the choice, right? And the choice C and D, that is vitamin D dependent rickets type 1 and type 2. The clinical features are almost the same, but the more severe, and that is a rare disorder, right? So correct choice for us is the B, that is X-linked hypophosphatemic rickets. Now next question. Question number 7. A person who had dinner at 8 p.m., right? Raat ko 8 baje he had the dinner, right? And gave his blood sample for glucose at the 7 a.m. almost after like 11 hours. What is the source of energy to the body during this period? So 11 hours is the gap. So body is deriving the energy, that is glucose, from where? Dietary glucose. Do you think dietary glucose survived this long? Not really. Up to 2 hours, 4 hours max. Hepatic gluconeogenesis. What goes first? We have these glycogen stores that we should be exhausting first, right? And hepatic glycogenolysis is the correct choice. And this, if you can remember from the class, the first is the fed state, absorptive stage, 2 to 4 hours after the meal. We are talking about 11 hours gap. 11 hours is where? Early fasting, 4 to 16 hours. That is what? Glycogenolysis. That is where? In the liver. That is where? In the liver. In the prolonged starvation, muscle proteolysis, that is the muscle. Right, we don't use, and the muscle glycogen stores are used by the muscles only, right, not by the entire body. So for us, the correct choice is what? The C, hepatic glycogenolysis for this question. Question number 8, female on maize as a staple diet. Okay, trivial question. History of necklace-like lesions. Maize, necklace-like lesions. Which of the following symptoms can be seen in this patient? What could be these lesions? Cazal's necklace. What do we expect? D. Maize is leading to, excessive maize is leading to the Pellegra niacin deficiency, right? That will lead to dementia. Very good, all of you. Diarrhea, dermatitis. It is not just on the neck, but it can be all over the body also, right? And then finally, it may lead to the death also. So choice A is the choice. Memory loss is usually because of vitamin B12 deficiency, right? B12 is more common in the older adults and the patients with the vegetarian, because the major source of B12 is usually like most of the meat, the eggs and all these, they contain the vitamin B12, right? And the red hair folliculitis is not the one. Bleeding gums is because of the vitamin C deficiency, right? That is scurvy we are talking about. So the correct choices for us is the choice A, that is diarrhea, dementia. Again, actually in this question paper, there were 2 questions on like revolving around the same topic. This is the female on the maize as a staple diet. There was another question in a patient on maize diet with DDD, diarrhea, dementia, dermatitis, which is of the following is deficient. So that is like you are getting 2 questions right if you know the if you know the concept, right? So that is vitamin B3, that is niacin, right, Vidhu? Very good. That is choice C. And now this is the entire list. You can take a look of all the vitamins and the deficiency disease that are related to them, right? Vitamin A, what you will see, the night blindness, right? Folate, megaloblastic anemia. Cobalamin, that is B12, B6 and B12, megaloblastic anemia you will see. Thiamine, we know about the beriberi, wet beriberi, dry beriberi, and then there is infantile beriberi also, because if the mother's milk is deficient, right, the baby who is feeding will get infantile beriberi. And then there is Wernicke-Korsakoff syndrome also. Sometimes they make it together, sometimes it is different. That is more of a confabulation kind of a thing that we see, right? Riboflavin is what? Non-specific fatigue, eye changes, dermatitis, lots of things. Niacin, pellagra, all the time we see. Vitamin B6 is also related to what? The dermatitis, neurological, but majorly we see it is in relation to the homocysteine levels and the megaloblastic anemia. Okay, so this is kind of a triad, you can see. This is kind of a triad, and that is also related to B12. If you can remember the homocysteine methionine relationship, we will discuss it a little bit towards the end of this video, right? C is what? C is C is the scurvy, right? D is rickets in children, osteomalacia in the adults, right? Now let's move to INI-CET July 2021 questions, right? There was this question about the microRNA binds to mRNA to which site to inhibit translation. See guys, so many times there is a confusion between all these things. You have miRNA, you have siRNA. This is all revolves around a gene therapy concept. Most of you have this is kind of a common thing, gene therapy. Okay, we have these genes which we are going to take into account to treat certain rare disorders, right? So either what we can do is if the gene is very active, we can down-regulate it. If the gene is not working like 100%, we can up-regulate it, okay? Sometimes gene is not, we don't need it, we can do the knockout of the gene, that is gene knockout. Sometimes the gene is missing, we can place a useful gene in to for the treatment, that is gene knock-in. So there are 4 things that are there, right? Knockdown, knock-up, gene knockout, down, up, in, out, right? 4 things are there. Whenever you are knocking anything out and bringing something new in, that is role of the CRISPR-Cas. CRISPR-Cas does this. CRISPR-Cas9 system does this. Whenever we are talking about the down regulation and up regulation of the gene is there, but we are trying to fine tune it like we do on the radio, we fine tune the channels on the FM, AM, right? So that is done by what? These gene regulatory system, miRNA, siRNA, right? And miRNA, they do what? They inhibit the gene transcription, or you can say they do the gene suppression. At which level? The transcriptional level. These are, you can say, the major broad terms. If you remember these terms, you will be able to make out the answer rather than knowing the entire theory behind it. Okay, so miRNA binds to mRNA. To which site to inhibit the translation? Right, here we are talking about the translation. So here is, first of all, know how we synthesize it, because that can be another question. So I have added this slide also. Initially, it is synthesized as pre-miRNA, and from pre-miRNA we get miRNA, that is the mature miRNA. Same way we synthesize the RNA and then we get the mRNA, that is a mature RNA. Same thing we do it, right? But this is a very important stepwise process. First we synthesize from the gene the pre-miRNA, right? And that looks like a hairpin loop kind of a structure, and that needed to be cleaved so that we can make a double-stranded precursor, right? That is called as pre-miRNA. We have to cleave it, and that is done by which? The dicer. right? So dicer does it, and then it is exported out. Who is exporting it out? The exportin. Name is easy. Exporting is exporting it out, right? And then it is going to take a risk. It is going to take a risk here. It is going to take a, not R-I-S-K, but risk. Who is going to take it to the targeted gene which is needed to be suppressed, right? So risk is helping the miRNA or mature miRNA to take it down to the targeted molecule. So you have miRNA which is ready with the risk to do the action. It acts where? It acts on the 3' UTR, untranslated region of the target gene which we want to suppress, which we want to down regulate. Okay, so the answer to this is choice C, 3' UTR. But I have told you dicer, exportin, the risk complex. You should be very familiar with the names also. They may ask you these things also. CRISPR-Cas9 they have already touched in the exam, so we should be knowing about the CRISPR-Cas9 system also. Right, now there was another question. What is the effect of RNA interference? Earlier we said miRNA, but there is this RNA interference. What is this? miRNA interference is a process, and who are the tools to interfere the miRNA? That is your miRNA and your siRNA, right? They interfere with the RNA. That is why they do it. And what they do it? Are they doing the knockout and knock-in? No, CRISPR-Cas is doing it. They are going to do the down regulation. Up regulation is done in very few cases. There are lots of researches, but really up regulation we have not done on humans so far. Down regulation is the most common process that we are doing. Gene knockout and gene knock-in by CRISPR-Cas9 system is a good tool. We have done, researchers have various success stories to tell, but still whether to use this system practically in the day-to-day life, because we are manipulating the genes, that is kind of an ethical issues. So that is why that is not available in the market for the treatment also. But researchers have done a lot of thing on the CRISPR-Cas system also, right? So gene knockout and knock-in is removal of the desirable gene, right? Knock-in is addition of the desirable gene, that is choice B, and that is done by CRISPR-Cas9 system. Here we are talking about RNA interference, right, which is also known as co-suppression, post-transcriptional gene silencing, that stands for PTGs, and the quelling. So other than this term, you can see any of these terms: co-suppression, post-transcriptional gene silencing, PTGs you can make it out, and the quelling are the similar, also known as AKA. And that does what? That does the gene knockdown, right? And this is This is a process. And the tools for this process is miRNA and siRNA. And they may ask you this another question related to this. What are the features? It is more precise, it is efficient, it is stable, and it is better than antisense therapy that is given for the gene suppression. Okay. There is this extra H thing. The miRNA and siRNA stands for microRNA and the small interfering RNA. And they are what? They are small ribonucleic acid. RNA is ribonucleic acid. Are they large or small? They are small ribonucleic acid, which is going to interfere with the RNA, right? And what they do is they prevent the translation via two processes, majorly via post-transcriptional gene silencing, and there are examples of the pre-transcriptional gene silencing also. So these are the key features, remember that. miRNA, siRNA are doing the RNA interference. They are what? They are very small in size, right? And they act either by the post-transcriptional, also by the pre-transcriptional message, gene silencing mechanism. If they give you both the choices, right, then pick the post-transcriptional gene silencing, because that is the major mechanism. Okay, and this is kind of a defense mechanism of the parasites also, like for the small microorganisms. Well, the correct answer is for this is the D, that is gene knockdown. Let's move further. Question number 3. Which of the following is enzymes and its, you are given an enzyme and its diagnostic marker? Is incorrectly paired. We have to find the incorrect pair. Lipase, acute pancreatitis is choice A. Right? In acute pancreatitis, what do we see? The serum lipase and amylase levels are elevated. So this is a correct choice. This is a correct pair actually, not the choice, but this is a correct pair. Ceruloplasmin hepatolenticular degeneration, right? Hepatolenticular degeneration is Wilson's disease that leads to the low ceruloplasmin level. So this pairing is also correct. C is beta glucocerebrosidase and von Gerke's disease. Beta glucocerebrosidase enzyme deficiency leads to what? Von Gerke disease? No, it is going to lead to the Gaucher disease, right? Gau is glue, right? And von Gerke's disease is because of the deficiency of G6 dehydrogenase, right? So this is incorrectly paired, and that what is being asked in the question, right? LDH1 to LDH2 ratio is related to myocardial infarction. Yes, we know that, right? Lactate dehydrogenase, right? These are the two isoforms of lactate dehydrogenase, LDH1, LDH2. LDH1 predominates in the heart, whereas LDH2 is majorly found in the blood. Under the normal condition, LDH2 concentrations are higher as compared to LDH1, but during the cardiac tissue damage such as MI, right, this ratio flips. The LDH1 is higher as compared to LDH2, and we call this as a flipped LDH ratio. Okay, so this is an extra edge point I wanted to give you out, but the incorrect pairing is this one, that is choice C. Dr. Kabir Rajput, yes, I can explain von Gerke, but maybe if I get time after this session or in another session, I'm definitely going to take a note and I'll explain you all the glycogen GSD disorders also, right? Insulin inhibits which of the following? Insulin inhibits which of the following? Insulin inhibits which of the following? Come on, A, B, C, D, HSL, LPL, gastric lipase, pancreatic lipase. Choice is hormone sensitive lipase. This is the figure I have used in the class, which is, what is this telling you? The epinephrine or glucagon. We are not talking about here the insulin, right? Insulin is released when it is in, the system is in fed state. But here we are talking about the fasting state, opposite to it, okay? Insulin and, epinephrine or the glucagon binds to this 7-TM receptor. Why this 7-TM receptor? 7 means there are 1, 2, 3, 4, 5, 6, 7, right? 7 transmembrane. This is the transmembrane regions, right? So 7 transmembrane regions, like it is going to talk to this, which is going to transfer the signal inside the cytoplasm. Then adenylate cyclase is activated, which is going to convert ATP into cyclic AMP, and the increased level of cyclic AMP is going to activate the protein kinase A. Protein kinase is going to do this. Kinase does what? They do the phosphorylation. So they are going to phosphorylate hormone sensitive lipase. So glucagon is doing what? Glucagon wants hormone sensitive lipase to be in the action, so that it is going to degrade the stored triglycerides into the free fatty acids. Then the energy will be obtained from these fatty acids via the beta oxidation, right? That is in the fasting state. So if the person eats, if let's say I was in the fasting state in the morning, now I have started eating. So that means I'm going to release insulin. So do I want HSL to be working? No, I don't want HSL to be working. So I have to do what? I have to do the dephosphorylation of what? Dephosphorylation of the hormone sensitive lipase enzyme, right? So it is going to dephosphorylate hormone sensitive lipase, right? That what insulin does. So insulin inhibits what? Hormone sensitive lipase. We cannot say Saurabh antagonist to insulin, but you just remember that insulin is going to activate the phosphatase enzyme, which is going to remove the phosphate from hormone sensitive lipase. So it is getting dephosphorylated, right? So correct choice is A. That is HSL, which is an adipocyte enzyme that cleaves the fatty acids from the intracellular triacylglycerol, and it is activated by phosphorylation. Insulin does the dephosphorylation, so it inactivates hormone sensitive lipase, right? LPL. LPL is another adipocyte enzymes, but it is releasing the fatty acids from the circulating lipoproteins, right? Gastric lipase is the acidic lipase secreted by the gastric chief cells of the fundic mucosa, that is choice C. And at optimum pH, some books they say 3 to 6, some books say 2 pH. So go for any pH, just remember it works at the highly acidic pH. Pancreatic lipase, name is suggesting secreted by the pancreas, right? And it is responsible for the hydrolysis of the fats, cholesterol esters and the fat soluble vitamins, right? So this is another important question from this question that may be asked. Now, cytochrome oxidase enzyme is inhibited by all of the following except cytochrome oxidase enzyme is inhibited. Where we are talking about electron transport chain, right? And cytochrome oxidase, what did I tell you? It is inhibited by, you will see most of the gases, the poisonous gases, they inhibit it. Remember this from the class? This is the first complex, that is NADH dehydrogenase. Question is asking you about cytochrome C oxidase, that is complex 4. It is inhibited by all the gases. It is not inhibited by the methane, right? So D is the choice. Methane does not inhibit this, but the hydrogen sulfide, carbon monoxide as well as the cyanide, they do. That is methane. Am I wrong? To me it is methane. Next question is, what does the graph represents? Michaelis-Menten kinetics, cooperative binding, allosteric activation, or the sigmoid curve? What do we have here? In the x-axis we have substrate concentration. On the y-axis we have the rate of the reaction, and this curve is indicating, right, as soon as we are increasing the substrate concentration, the rate of the reaction goes on increasing and it reaches to a maximum level, right? So this is kind of a hyperbolic curve, and this is V max that we indicate. So this is what? Ganesh, cyanide is also a gas. Methane is also a gas, but cyanide is more poisonous. It inhibits. that is a general term that I have told you, that the gases, they inhibit the cytochrome oxidase. All the given ones are, we are talking about the gases. Methane does not inhibit the cytochrome oxidase, right? That is like, exceptions are always there in the exam. So just remember that, Ganesh, okay? Thank you. Yeah. So coming to this point, so this is a hyperbolic curve. Do we see this in Michaelis-Menten kinetics, cooperative binding, allosteric activation? Cooperative binding, where do we see? We see it in the hemoglobin, and that gives rise to what? This red colored cooperative curve, that is the sigmoidal curve. So technically B and D choices are kind of a same kind of a choices, because cooperative binding we are see the sigmoidal curve, and here there is this sharp increase, right? There is a sharp increase here in this curve. Let's not go in detail, otherwise we will not be able to cover all the question. Or the allosteric activation. Allosteric activation is also an sigmoidal curve, that is S curve, right? So which is this? This is very obvious. The Michaelis-Menten kinetics, that is the choice A, that as more you add the substrate concentration, there will be an increase in the rate of the reaction, because the substrates are available for the enzymes. But soon after some time, all the enzymes, see, enzyme concentration we are keeping constant, right? So if one enzyme is binding to one substrate, so initially let's say you have 100 of enzymes and you have 10 substrate there. So 10 substrates are binding to 10 enzymes, and rest 90 are free. So if you add 100, so all enzymes and substrates, they are busy in mingling with each other. So no matter how much substrate you are adding, all enzymes are occupied with the substrate. So none of the girlfriend they want, because they already have one of the girlfriend as a substrate. So the velocity reaches to a maximum level, that is Vmax. So that is Michaelis-Menten kinetics, that is choice A is the correct answer to this question. In fasting RBCs, very simple, very easy question. RBC always use glucose. Fed, fasting, prolonged fasting, starvation, it cannot use anything because it is lacking the mitochondria, right? So the choice is A, right? Increased methyltetrahydrofolate accumulation occur in deficiency of which enzyme? Very heavy question. Methyltetrahydrofolate, they may write you this. We will take all the options one by one, right? Folate levels. First of all, who is the marker? Figlu is the marker. Another important question for the exam, right? Not the recall, but they may ask you, right? The Figlu is the marker of the intracellular folate levels. And if the folate levels are low during the pregnancy, we know it is going to lead to the NTD, neural tube defect, right? And deficiency of either the folic acid or the vitamin B12, either both are absent or one of them are absent, right? That is going to lead to what? The functional folate deficiency. What is functional folate? Functional folate is folic acid in active form. What is the active form of the folic acid? Methyltetrahydrofolate, right? That will affect the dividing cells. Why? Because where do we need? For the DNA synthesis, we need the folate that we have discussed in the molecular biology also, right? Also, it affects the bone marrow and it also leads to megaloblastic anemia. This is another well debated questions. If the people who take folic acid in excess in the diet, that also is the dangerous. You can pause this and you can read it maybe later. Now, choice B was B6, that is pyridoxal, right? That is activated form of vitamin B6, PLP, pyridoxal phosphate is the active form. And what do we see in the patient, right? It is first of all very coenzyme for glycogen phosphorylase, right? This is also a high yield, you can say a key point, you should be noting it down. That is glycogen phosphorylase. And its deficiency symptoms are these: sideroblastic anemia, peripheral neuropathy, homocystinuria, oxaluria, and xanthinuria. Right? The question was asking about the methyltetrahydrofolate accumulation. Is it B6? We don't see it in B6. B9, B12, B1. What do we know about B1? This is not the choice. B1 is what? Is a thiamine deficiency, that is the beriberi, all kind of a berries, right? So this is not the choice. So B9 or B12, what is the choice? Yes, Ganesh, this is the methyl donor. Another methyl donor is SAM, S-adenosyl methionine, okay? So that is, there are two methyl donors. One is this, second is this. So which one is the choice, A or C, B9 or B12? Am I right? I have cleared this in class also, that homocysteine to methionine, what we are adding, meth is added, right? So it means we are adding a methyl group that is highlighted here in the blue. An enzyme involved is what? Since we are synthesizing methionine, enzyme involved is methionine synthase enzyme. Methionine synthase enzyme needs B12. So if there is no B12, there will be no synthesis of the methionine. And then what will happen? Who is donating this methyl group? This methyl group is donated by methyltetrahydrofolate, right? So if B12 is deficient, there will be an increased level of methyltetrahydrofolate. And this is also known as what? The folate trap. That is a folate trap. And which one? This is not vitamin B9. Vitamin B9 we see afterwards. This is B12. Okay? Very good. And there are other two enzymes. Note these points also. Methyl malonyl coenzyme A mutase and methionine synthesis, I told you. These are the two enzymes which requires vitamin B12. Remember, methyl malonyl acid urea is also because of vitamin B12 deficiency. So two things are increased in B12 deficiency. Majorly we see methyl malonyl acid urea, not so commonly asked, but folate trap, or you can say increased methyl tetrahydrofolate is most commonly asked. Why we call it a folate trap? See, the folate is this. Since B12 is not working, we are not able to convert methyl tetrahydro to folate. So all the folates will be trapped as a methyl tetrahydrofolate. So that is a folate trap, because going to this is working, but the methyl 2 tetrahydrofolate is not working. So all the folate will be trapped there as a methyl tetrahydrofolate. Okay. And the deficiency causes, what do we see? The pernicious anemia or the functional folate deficiency or folate trap. These are the keywords for the exams I'm talking about. Okay. B1 thiamine deficiency is not the case, but in the previous my doubt solving question, one of the student, he was asking about the thiamine, right? Thiamine works as a coenzyme for pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, transketolase and beta keto acid dehydrogenase. These are the majorly 4 commonly asked enzymes which are using TPP in the which are using TPP as their coenzyme, okay, right? And the deficiency leads to the beriberi, right? The dry beriberi. These are the features, kind of a dry features. Then the wet beriberi, it is more or less related to the CVS, right? We see the edema, palpitation, cardiac tachycardia, high through output cardiac failure, right? And then one of you asked about the infantile beriberi, right? As I have told you previously in this video also, whose mother is deficient in vitamin B1, Wernicke's encephalopathy, it is related to global confusion and ophthalmoplegia. Okay. Now next question is, which of the following method cannot be used, for protein precipitation? Adding alcohol and acetone? Yes, it does. Moving away from isoelectric pH. Isoelectric pH is the pH where there is net charge on the protein is 0. So either you move it, can you move it if the charge is 0? You cannot move it in either way. It is fixed there. So that is the point when you can precipitate the protein out, because it is not moving. So if you move the pH, if you shift the pH towards the acidic or basic, then protein is going to get moved. So that is against the precipitation. So this is the correct choice. Adding trichloroacetic acid, using heavy metal ions, A, choice C, choice D, all are the ways for the protein precipitation, right? So B is not the choice for the protein precipitation. Okay, and I'm listing again that we have discussed. We can precipitate the protein based upon the solubility, that is the crude separation, right? How do we do it? By adding the heavy metals. Salting out is the process of precipitation. This is a process of a precipitation, right? We can add the alkaloid reagents like trichloroacetic acid, phosphotungstic acid, sulfosalicylic acid, also by the dehydration process, also by adding the organic solvents such as ether and the alcohol, right? Neutral salts is like we can have something associated with the ammonium sulfate, right? And this was based upon the solubility criteria. But we can separate the proteins based upon the molecular size, molecular charge and affinity binding also. Molecular charge is like dialysis that we do, ultracentrifugation. What is the basis of dialysis? Proteins are separated based upon the molecular size. That is the another way to look at it. Okay, gel filtration, chromatography, these are separating the proteins based upon the molecular sizes. Molecular charges means we are talking about the charge. So ion exchange. High performance liquid chromatography, HPLC, right? Electrophoresis, isoelectric focusing, affinity binding is affinity chromatography, precipitation with the antibodies also. Okay, so these are the various ways we can separate the proteins. Now, INI-CET November 2021 questions. Most preferred chromatography of the glycated hemoglobin. This is glycated. One of you have asked me whether it is a glycosylation or glycation. This is glycation. They may write glycosylation. In a way, it is also correct, but glycation is most correct term. We can do all the methods. We are using all the methods that are available in the market, A, B, C, D. So this was a very tricky question, how to pick the best answer. So can any one of you tell me what is the best answer? All the methods are available in the market, even the enzymatic, right? The correct, better preferred choice is ion exchange chromatography. The best reason to find it out, what I have digged out, is because it is positively charged. So ion exchange means we are working with an ion, so it is like preferred method. But all methods are used. All methods are used, to tell you the truth. Very good. Ganesh, Robin, Zahra, Shubham. You all seem to be very well prepared for the upcoming exam. Which of following is not glucogenic? Another very simple, straightforward. Glucogenic or ketogenic? Glucogenic, you will see the pyruvate, lactate, oxaloacetate, right? All this is glucogenic. Not is the acetyl coenzyme, acetoacetyl coenzyme, beta hydroxybutyric acid, like the ketone bodies if it is forming, so it is not glucogenic, right? Right, so enzyme choice A is your correct answer. Very straightforward, simple question. Thymine is a coenzyme for. Third time we are seeing this. I have just given you the entire list. 4 enzymes were there for the TPP, right? Thymine coenzyme form is TPP. Sometimes they write it TDP, thymine pyrophosphate or thymine diphosphate, one and the same thing, right? Is for what? A, B, C, D, PDH, pyruvate carboxylase, PEP, CK, PEP, CK, pyruvate kinase, PK. It is PEP-CK, PK, PC, PDH. What is this? Exactly, right. A, pyruvate dehydrogenase, right? This is, I have just shown you previously. These are the 4 enzymes TPP plays around with. So TPP is having 4 boyfriends, right? PDH, alpha KDH, TK and BCKDH, right? These are the 4 ones. So PDH is enzyme related to what? The link reaction. Another very important question that may ask you in the exam. Pyruvate dehydrogenase is link reaction, right? And it requires 5 of the coenzymes and the cofactors. Lipoic acid it requires thiamine also, it requires the nicotinic acid. So, and it is linking the glycolysis to the tricarboxylic acid, and there is a decarboxylation step that is going on also, that is removing the carbon dioxide, right? So this is this. Pyruvate carboxylase B. Why it is not choice? This requires biotin as a cofactor, right? All the carboxylase, be it a pyruvate carboxylase, propionyl coenzyme A carboxylase, methyl crotonyl coenzyme A carboxylase, what do they have in common? The carboxylase, they require biotin. Okay. Choice C, PepCK, what it requires? PepCK requires manganese ion, right? So it is not the choice for us also. So this is PepCK. Where do you see the PepCK? This is this enzyme. This is doing what? This is converting oxaloacetate to the phosphoenol pyruvate, which will go to the gluconeogenesis. So it is activated in the gluconeogenesis and it requires manganese ion. Pyruvate kinase converts what? Phosphoenol pyruvate to the pyruvate. That is kinase. It can be doing either way, right? It is key enzyme for the glycolysis, produces 2 ATP and it requires magnesium ion. And this step is, another property for this step is, this is substrate level phosphorylation. And deficiency leads to hemolytic anemia. Few extra H points for you guys. CRISPR-Cas9 repair method is associated with what? Remember I told you now they are touching the CRISPR-Cas9, so that here comes a new topic that you should be learning for the examination. Non-homologous end joining repair, also known as NEHG repair, mismatched repair, nucleotide excision repair, base excision repair. Here we are dealing, in all the 3 cases, we are dealing majorly with the single strand. Mismatch is what? There are these 2 strands. So here there is this one base which is not correctly base paired with the another one. So you are going to chop this off and repair it. Nucleotide excision repair, right? Or the base excision. Nucleotide, you remove the entire nucleotide and put the entire nucleotide, correct one. Base is you are just removing the base and you are putting the correct base in at the place, right? So CRISPR-Cas method is associated with, I'm going to like briefly give you the overview. This is the CRISPR-Cas9 typical model from the Harper, right? What it has, this here, look at the back. Do you see this 8 kind of a ribosome or 8 kind of a structure? What is this? This is your Cas enzyme. This is your Cas9 enzyme, right? So CRISPR-Cas is Cas9 enzyme is there, that is indicated here in this yellow shady thing. And then you see this green guide RNA, which is again composed of 2 pieces, which I am not discussing here, that they together come to the target sequence, right? Where we want to knock out the gene by chopping it off, and that is a double-stranded chopping, okay? That is a double-stranded chopping, that is cleaved right here and the cleaved right here. More precisely, this is an elaborate version, the Cas9 nuclease, and this guided RNA comes from the CRISPR. They come together, they target the sequence, right? And then they chop it off. And that is double-stranded DNA break. So CRISPR-Cas9 system does what? Double-stranded DNA break, right? And that is repaired by the non-homologous end joining repair or homology-directed DNA repair. Choice has given us this, so this is the correct choice. This is the best choice actually out of the homology-directed DNA repair, because homology-directed DNA repair is less correct. More accuracy is with the NEHG, that is non-homologous end joining repair. Okay, so this is the one you should be knowing about the full form of the CRISPR. That is funny, right? So you should be knowing about this, and now you have a brief idea about how the CRISPR is working. CRISPR-Cas9. CRISPR is a sequence. Cas9 is an enzyme that comes together, and how we have like figured it out, this is kind of an defense mechanism for the bacteria. Let's say any pathogen invades the bacteria, right? What the bacteria does is it has a memory. So all the infections it has gone through in the during the lifetime of that bacteria, right? It keeps it in the memory. It keeps a piece of that infecting bacteria, kind of infecting like a pathogen inside its brain in the sequence. As soon as the same pathogen comes in, the CRISPR system goes and checks for the sequence whether they have the same kind of infection before or not. If it is there, it is going to call the Cas9. Okay, nucleus come in, we have the same intruder who is going to infect me, right? Because I already have this thing in the memory, and that is going to cleave the infecting pathogen. Okay, so it is related to the NEH, which requires maximum energy of assimilation. What do we mean by assimilation? Assimilation efficiency is A is I minus E divided by I minus E divided by I times percent gives you assimilation efficiency. I is the ingestion rate and E is deficiency or egestion rate, right? Do we have to know the math? Not really. We have to know a theme around it. We know what? We know the specific dynamic action, also known as thermogenic effect of the food. The specific dynamic action is associated with how we are processing and how we are assimilating the energy, right? So if we know the specific dynamic action of a particular thing, be it a carb or a lipid or a protein, we can say, okay, we know the assimilation maximum energy is required, like whether it is requiring a high amount of energy or a low amount of energy. For the specific dynamic action, what we know is the proteins or the amino acids, they account for 70 to 80% of the SDA, that is specific dynamic action, that we have discussed via this table in the class also, right? This is TEF, thermogenic effect of the food, also known as SDA. It is one and the same thing. We don't have to go in maths, right? And we know the proteins, they have the highest SDA. So it means the assimilation will be the high for the proteins as compared to the others. So answer to this is the protein. But they may trick you. Free amino acids or the polypeptides. So remember, free amino acids have the less assimilation as compared to the polypeptide form. Okay. What is true about mitochondrial DNA? Higher chances of mutation as compared to nuclear genome. That is true because they lack repair mechanism. Are they single-stranded circular DNA? They are double-stranded circular DNA. So this is false. Do they encode all the electron transport chain proteins? No, they don't. No. It has around 1600 base pairs, around 1600 base pairs. So this is what from our notes for the mitochondrial DNA, double-stranded circular. So choice 2 is false, right? Small, around 1600 base pairs. Exact number is 16569. Okay. Are they encoding the all chains? They encode the 13 proteins out of 67. So approximately 20 proteins. So the statement given to you, the 3 is false, right? So what is the choice? How many things we have correct? We have first and 4, that is correct, right? So answer to us is the first and 4. If they have not used the word around, then this also would have been an incorrect choice. They are using approximately 1600. Okay. Wobble hypothesis is due to which property of the codon? Wobble hypothesis. From the notes again, right? Genetic code is triplet code. It is degenerate. It is unambiguous, non-overlapping, non-punctuated, and the universal. This was another one of the question asked in the exam also. But here they are asking about what? The Wobble hypothesis, right? Wobble hypothesis is because of what? It explains the degeneracy of the genetic code. What do we mean by degeneracy of the genetic codes? We know there are 61 codons. 61 codons means there must be 61 tRNAs, but we don't have 61 tRNAs, right? That is explained by the Crick in 1996 that there is a degeneracy in the codons, and we will take a look into that. But before jumping into that, make a note for this point. Except the tryptophan and the methionine, more than one codon direct the synthesis of an amino acid, right? This is a very important point for you to notice, okay? So this is the Wobble hypothesis. What it is, here is your anticodon. Anticodon is always with the tRNA, and this is your codon. Normally, G base pairs with the C to code for the leucine. But according to the Crick, based upon the Wobble hypothesis, it is saying that G also can base pair with the U also for the same is leucine. Now you have to pay attention which base we are talking in the codon and in the anticodon. So this is codon. Codon is 1, 2 and 3. And anticodon is 1, 2 and 3. Opposite direction they base pair. So if this is 5:3, the anticodon is 5' to 3' this direction. So this is the first position in the anticodon can be variable. It is allowed. And this is the table for that. The anticodon first base U can base pair with either adenine or the guanine. The anticodon in the tRNA, if it has C, it is allowed to base pair with the guanine. If it has adenine, it is allowed to base pair with the uracil. If it has G, it is allowed to base pair with the uracil as well as the cytosine also. If it has inosine, inosine is similar to the guanine, it can base pair with any of these, that is U, C and A. That is the Wobble hypothesis, okay? So it is explaining the degeneracy of the code, okay? NEET PG 2020 questions. What if the percentage of the thymine residue in DNA is 28%? What is the percentage of the cytosine? Very simple thing. Thymine is 28. What we know is thymine base pairs with what? A base pairs with T, G base pairs with the C. If T is 28, so A will be what? 28. So it will 56 are this. Total is 100, right? So 100 minus 56 is what? 44. So means 44 are G plus C's. They should be equal. So 44 divided 2 is 22. So it is cytosine will be 22%. It's simple as that. The A should be equal to T, G should be equal to C. If T is this, if T is 28, A is 28. Total is 56. So we are left with only 44. So 44 has to be distributed equally between G and C's. ठीक है ना? So that is 22 of each. Patient complains of knee pain. Routine investigations are unremarkable and still the patient is unsatisfied. Same kind of a question we have seen before also. Urine turns black on standing. Which enzyme is involved? So what are the choices? Homogentisic oxidase, xanthine oxidase, tyrosine transaminase 4, fumarate acetoacid. They have changed the choices, but the same question is the repetition. Here now they are not giving the dehydrogenase, they are giving the homogentisic oxidase, right? Non-competitive inhibition has a what? NC has what? Normal Km. Normal Km with increased or decreased Vmax, or decreased Km with increased or decreased Vmax. What is the correct choice? This is the Michaelis-Menten thing, right? This is the Vmax. This is half of the Vmax, and if we drop it down on the x-axis, that is substrate concentration. It gives us the Km. So Km is the substrate concentration when it is half of the maximum velocity. Okay, and this is the chart we know. Michaelis-Menten plot, double reciprocal plot. Just mug this up. For the competitive inhibition, what do we know? We know is Vmax is unchanged, Km is increased. Question is asking you about non-competitive. Non-competitive is Vmax is decreased and the Km is unchanged. So what is the choice? That is B, normal Km and the decreased Vmax. That is non-competitive inhibition. Female on the maize diet as a staple diet, diarrhea, lesions in the neck region, right? We know that is because of the Pellegra is there, niacin deficiency. So A is the choice. Replacing alanine by which amino acid will increase the UV absorption protein at 280 nanometer wavelength? We know most of the amino acid solution, protein solutions, they are colorless. So, but there are these 3 branched chain amino acids, that is Phe, Tyre and Trip, right? They show the absorbance, and if you can remember this from the class, tryptophan absorbs the most around the 280 nanometer. So if we replace the alanine by the tryptophan, that is going to show the absorbance at the 280 nanometer. So after tryptophan we have the tyrosine, and phenylalanine shows the least. So tryptophan is what? It has what kind of a system? 6-membered fused with the 5-membered ring, right? Indole kind of a system it has. So a choice is A. Apolipoprotein B48 is an example of which of the following mechanism? Originally, we know the there is ApoB-100. Originally, ApoB-100 is there. That is made by ApoB gene. GTT, when we are going to do the transcription, we will get the CAA. But there is this mechanism, there is what mechanism? Kind of an editing. Right, there is a mechanism kind of an editing of what? Editing of RNA we are doing, in which there is a deamination done by the system itself. It is predefined. Cytosine is deaminated to give you the uracil. So rather than CAA, it becomes UAA, which is one of the stop codon, and the translation, it stops at the 48th position. So you get ApoB-48 rather than getting the normal ApoB-100, right? So this is what we are doing. We are doing the RNA editing. What else we know about ApoB-48? There is very important thing. ApoB-48 is what? Exclusive of chylomicrons. When we did the lipoproteins, remember? ApoB-48 is exclusively found on the chylomicrons, right? And ApoB-100 is related to the lipoprotein lipase, right, from which we can remove the fatty acid. Okay. Which among the following are the insoluble dietary fibers? See, hemicellulose, gums and the lignins. They both are insoluble dietary fibers, right? But the correct choice for this is the gums and the lignins, right? They are more insoluble as compared to the hemicellulose ones. Insulin-glucagon ratio is decreased. Which of the following enzyme will be most active? So if I place like this, insulin to glucagon, this is numerator. Childish math if we do. Denominator, just I'll write this, denominator. Okay. If this ratio is decreased, when this ratio will decrease? When the denominator will be a bigger number. If this is a bigger number, this is a smaller number, right? And if you divide a smaller number by a bigger number, then you will get a very small number as a result. So it means the insulin-glucagon ratio is decreased, right? When you will have the denominator, means the glucagon, that is we are talking about the starvation. In the starvation, which enzyme is most active? right? Simple as that. That is another way to look at it, right? Low IgR is stimulating, right, the mobilization of the stored nutrients because we are in the fasting state. Fasting state, we do more of the glycogenolysis, then the gluconeogenesis, and we break down the free fatty acids and the glycerol. And which enzyme will be activated? Glucose 6 phosphatase will be activated. This is another way to look at it and the simpler way to look at it, because most of the times in the exam we are confused between the names of the enzymes. They look very similar, right? Even at the birth, glucose 6 phosphatase activity is increased. Okay. Same, which vitamin deficiency? That is D, vitamin B3, choice D, right? Which of the following drugs act by inhibiting the DNA replication? 6-mercaptopurine is an analog of adenine and the guanine. They are the purines, right? Purines, what do we say always in the class? Purines is and the pyrimidines are the two things we talk about. Pyrimidine has a big name, so small structure, single ring we see. Purine is a small name, so big structures we see the two rings. So adenine and the guanine, the analogous is 6-mercaptopurine. So if we look at the pyrimidine synthesis, right, initially what will be, this is the PRPP amidotransferase, make the phosphoribosyl amine, and there are so many steps going on. Finally, we make the hypoxanthine. From the hypoxanthine, we make the GMP and the AMP. So here this 6-mercaptopurine, right, is converted into an nucleotide which seems similar to the adenine or the guanine, and it is getting incorporated. So it is affecting what? It is affecting the DNA replication. So A is the correct choice, right? A is the correct choice. And the conversion of the 6-mercaptopurine to a similar looking nucleotide is done by HGPRT first, and then PRP is going to incorporate into the elongating DNA strand. There is this allopurinol. Allopurinol is a prodrug for treatment for the gout, does the same thing. It acts the same thing, same mechanism of action you can say, okay. So choice A is the correct choice for us. Actinomycin D is a transcription inhibitor, right? It binds to the transcription initiation complex, right? And it prevents the RNA polymerase elongation. Mitomycin C interferes with the cell division. Asparaginase, it is used to treat the ALL, acute lymphocytic leukemia. Asparaginase work is it converts the asparagine and release the ammonia so that energy can be released from it, right? So if this is asparaginase is not working, so it starves the cell to death. So hence it is used in the treatment for the cancer. Which of the following drugs by inhibiting the transcription of DNA to RNA? DNA to RNA, rifampicin, nirofurantoin, ciprofloxacin, novobiocin. Answer is rifampicin, right? Rifampicin inhibits the DNA dependent RNA polymerase, right? And that enzyme is doing the DNA transcription. So you are not making the RNA from the DNA. Nitrofurantoin, right? It is involved as it is a DNA inhibitor. RNA inhibitor also interferes with the cell wall protein synthesis, right? It is majorly used in the UTI infections. Ciprofloxacin, it is a second generation fluoroquinolones, right? It affects both the gram positive and the gram negative, and it acts with the DNA gyrase or topoisomerase 2 and 4 you can say. Novobiocin inhibits the DNA replication, right? I'm sorry for the ciprofloxacin, I said it acts with the DNA gyrase. Mistakenly I did that. It is novobiocin which inhibits the DNA gyrase, okay. As per NCEP ATP3, which is not included in the metabolic syndrome. What is NCEP ATP3? NCEP is National Cholesterol Education Program, right? There is this panel for the adults, right? And that is related to the high blood cholesterol in the adults. This is about the testing of the high blood cholesterol in the adults, that there is this panel, we call it as NCEP ATP3, right? NCEP is National Cholesterol Education Program for the Adult Treatment Panel 3, we call it, right? And this panel has made a particular criteria to diagnose this metabolic syndrome, that is the high blood cholesterol. And what are those diagnostic criteria? Increased waist circumference, that is central obesity, so this is included in the criteria. Elevated triglyceride, that is hypertriglyceridemia, that is also included. Low HDL cholesterol, but the question is asking about the high LDL, so this is not included. The fourth is the hypertension, this is also included. So answer to this is choice B, that is high LDL. Perfect. Impaired fasting glucose more than 110 mg per deciliter is also included in this criteria. So there are 5 criteria for the NCEP ATP3. Okay. True about protein structure. What is true about protein structure? Secondary structure is decided by amino acid sequence. Tertiary structure is responsible for stability is the correct choice. Secondary structure is not decided by the amino acid sequence. Sulfide bond is present in tertiary structure of the protein. Do you see it? Tertiary structure of the protein. Primary structure is lost on heating or denaturation. It is tertiary structure which is lost on heating and the denaturation. Correct choice is B. Okay. Serum bilirubin can be measured by what? Serum bilirubin can be measured by a van den Berg reaction. Right, what is bilirubin? Bilirubin is a yellow compound excreted through the bile and the urine, and elevated levels, they are indicative of the disease. Right, what do we see? The yellow discoloration in as the jaundice, right? And this is one of the pathway how the heme is degraded, that we have covered under heme degradation in the class, right? So heme is converted to biliverdin, that is a green colored compound, right? In the birds, they excrete the green colored feces, that is the biliverdin, but in humans, in us, we convert it into bilirubin, which is again the conjugated bilirubin, then via the bile duct, right? Via the bile duct, it is converted into the stercobilinogen. Stercobilinogen imparts the brown color to the feces. Then it is moved right into the kidneys, where it is converted into the urobilinogen also, that is imparting the yellow color to the urine. So that was entire thing that we have discussed in the class, and this is the table. If you can remember, the reaction, van den Berg reaction, component detected is the serum bilirubin. For the Ehrlich, that is the one choice, we detect urobilinogen, right? For the Scheck-Singers, we detect the urobilin, where the Fauchett's, we do the bile pigment, and this is the urine, and here is the serum, right? So answer to this is the choice A, that is serum bilirubin is measured by van den Berg reaction. INI-CET November 2020 questions. All are true regarding telomerase, except telomerase is the name of enzyme which synthesize telomers. Telomers are what? Telomers are the ends of the chromosomes, right? So telomers are the ends of the chromosomes. You find it here, you find it here, right? This is a nice chromosome, right? Centromere is this, and these are the sister chromatids which are the same, but if there is a gene which is on the same chromatid but they are identical, we call them as an allele, right? So coming back to the telomers, do they have reverse transcriptase activities? Yes. Only found in eukaryote? Yes. They maintain the chromosome length? Yes. Responsible for maintenance of DNA replication? No, right? So this is the choice which is incorrect. Ends of the chromosome contain the structures called telomeres. Telomeres contains of these are other features. You may see these as the options in the exam also. So I thought to incorporate it. Telomeres consist of short TG rich repeats, right? It's like T, T, A, G, G, G. The sequence is similarly like this, but they can extend to the several kilobases, right? Telomerase is what? It is a multi-subunit RNA template which contains what? RNA-dependent DNA polymerase. So, RNA-dependent DNA polymerase means it is a reverse transcriptase, okay, responsible for the telomere synthesis. Thus helps to maintain the length of the telomere. What do we know about if the telomere length gets decreased? With the aging, telomere length gets decreased. So cancerous cells, what they do is they keep this telomerase enzyme very active. So the telomere length is always maintained. So they survive for the longer period of the time, like amar ho gaye hain. Telomere shortening has been associated with both malignant and transforming and the aging, right? Cancer cells, they choose the telomeres as their saviors so that they can survive for a longer period of a time. So targeting telomerase enzyme for treating the cancer is one of the therapy that we use, okay? DNA methylation is not involved in what? Chromatin remodeling, RNA splicing, gene silencing, base excision repair, what? What do we know about DNA methylation? It is involved in the gene silencing, X chromosome inactivation and establishment and the maintenance of the imprinted genes, suppression of the viral genes also, and the carcinogenesis also, right? So 2 and 4 are the correct choice, right? Because they are not involved. RNA splicing and the base excision repair is not involved. Like DNA methylation don't have to anything do with this. The correct order of the steps in PCR reaction, I have a very good day. D is a denaturation, right? You can call it as a dawn time. Early morning time is a dawn time. A is afternoon. A stands for annealing. E is the evening. That is extension. So C is the correct steps, right? Post meal with high thermic effect is seen in, right? H, high thermic effect, same specific dynamic action, right, that we have just before seen, right? Specific dynamic action, also known as thermic effect of the food, also known as the dietary induced thermogenesis, right? It is what? It is the amount of energy expenditure above the basal metabolic rate. And who has this? The protein is the one which are having high thermogenic effect of the food, right? That is choice B is the correct answer. That is protein, okay? Same as the assimilation question that we have done before. There is the alternate splicing of the gene which have intron segment 1, 2, 3, 4, 5. And you have the primers W, X, Y, Z which yields 2 products, and we are interested in getting the product A. What is the best thing about the product A? What we know is it has 1, it has 3, it has 4, it has 5. What it do not have? It do not have 2, right? And these are our choices. So W we have to use no matter what. Now we have to decide whether we want the X, Y, Z primer or XY primer, right? So X is related to segment 2. Do we need segment 2? No. So we don't need X. So this choice is gone, this choice is gone. Y and Z are left. Now look at this. If we choose Y, that is going to go all the way down. So do we need Z? Because Y is going to cover Z anyways, right? If we don't have Z, then Y is going to go all the way around. You will have get the product, that is 1, 3, 4, 5, because 1 W is going to give you the 1 and Y is going to give you 3, 4, 5 all. So answer to this is the B, that is W and the Y, because you don't need 2, so you don't need X. Y can do the work of Z primer, that is a kind of a forward primer. Y can do all of it, it can cover the Z also. We don't need Z also, why to use 2 primers? Normality of 2 molar HCl is dash as 1 molar H2SO4. Good lucky for us is they are giving the molarity. They are not giving the normality and the molarity. Then you have to convert. So if you have HCl, HCl is going to furnish 1 proton. H2SO is going to furnish 2 protons. You have 2 molar. 2 molar HCl will give you what? 2 protons. 1 molar H2SO4 will give you one of the 2 protons. If you look at it, how many protons you are getting there? You are getting 2 protons each. So it is same. It is same, right? Enzyme with highest catalytic efficiency is, what is catalytic efficiency? Catalytic efficiency is Kcat over Km. So look at all the numbers, they are giving you the Km. Here is the micromole, nanomole, micromole, 4 micromole, and the second units are the same, that is cat. So if I write Kcat, just by looking at the number, the highest catalytic efficiency means the whatever is the answer, we want a big number. When you will get a big number? When the denominator is small. When the denominator is small, then if you will divide it, you will get a bigger number. So Km should be small. Where is the Km? That is, what is the Km? That is the smallest number. Do you think this is the smallest number? And Kcat is the biggest number? Do you think? Just look at the mathematical expression, look at the numbers, don't look at anything else. That is a choice. That will give you the bigger. You can do maths for all of them. Which of the following enzymes are involved in gluconeogenesis? Pyruvate carboxylase, phosphoenol pyruvate carboxykinase, hexokinase, pyruvate kinase, glucose-6-phosphatase. In gluconeogenesis, what we have? We have PC, we have Pepsi-K, we have fructose 1,6-bisphosphatase, we have glucose-6-phosphatase, right? We have this, A and 2 we have. So answer to this is, 5 also we had, is glucose-6-phosphatase, yes. This was the choice given. My apologies for that. So choice is A, 1, 2 and 5. Which of the following mechanism occur in the patient with type 1 diabetes mellitus with low insulin contributing to ketogenesis and ketonuria? Low insulin contributing to ketogenesis and ketonuria. How do you get the ketonuria in ketogenesis? Low insulin means what we are doing. We are talking about the fasting. Glucagon is up. Glucagon up. It means is it going to decrease the fatty acid synthesis? No. Is it going to increase the mobilization of fatty acid? Yes. Is it going to increase the shunting of acetyl-CoA to the TCA cycle? Is it going to do the shunting? Acetyl-CoA to TCA cycle will be shunted to give you ATP. What about the glucose that you need? Partly right, but not exactly right. Down regulation of beta hydroxybutyrate synthase system. So it means, are we interfering with the ketone bodies formation that takes place during the fatty acid oxidation, right? So low insulin means increased HSL, hormone sensitive lipase, that we have discussed before. So it means there is increased lipolysis, that is the increased mobilization of fatty acids from the adipose tissue. So choice 2 is the correct choice. 3 is a partially correct. It is not related to directly the insulin, right? Low insulin is going to do the increased mobilization of the fatty acid, because low insulin is an indicator of the increased glucagon level. Okay. Which of the following enzyme requires B12 as a coenzyme? I have discussed this before also. There are 2 enzymes which requires vitamin B12. Methylmalonyl coenzyme A mutase, B12 is deficient. It is going to give rise to methylmalonyl aciduria, methionine synthase. If it does not have the B12, it do not see, that is going to lead to the folate trap, right? And these are the B12, where B12 is also seen with the ribonucleotide reductase, not the folate reductase, leucine aminomutase also, right? So the answer to this is 1 and 2, right? B12 is happy with the MM, right? Both mummies, it is happy with the methylmalonyl and the methionine. B12 is happy to go. AIMS June 2020, which of the following is not an enzyme in cholesterol biosynthesis? This is the cholesterol biosynthesis table we have done. It has a thiolase enzyme. It has HMG-CoA synthase enzyme. It has HMG-CoA reductase enzyme. What it does not have? It does not have HMG-CoA lyase enzyme, right? So this is the answer. Where do we see HMG-CoA lyase enzyme? HMG-CoA lyase enzyme is seen with the formation of the ketone bodies. This acetoacetate is a primary ketone bodies. Acetone and beta hydroxybutyrate is a secondary ketone bodies, right? Enzymes are same. But remember, this is cholesterol synthesis, right? And HMG-CoA synthase, this is what? This is taking place in the cytoplasm. Cholesterol, cytoplasm. So isoenzyme is in the cytoplasm, whereas in this HMG-CoA synthase, right, where this ketone bodies is taking place in the mitochondria. Oxidation is taking place in the mitochondria. Which pathway describes the synthesis of metabolically active vitamin D? That is calcitriol we talk about, right? And I always show you this, and I always tell you to remember the silk. Silk is what? You have the skin, you have dehydrocholesterol. On exposure to UV light, it will convert into cholecalciferol, right, which is an inactive form of vitamin D3. Then it is transferred to the liver, then it is transferred to the kidney, and finally upon the action of 1-alpha hydroxylase, we get calcitriol or 1,25-DHCC, which is an activated form of vitamin D. So the process is silk, S-L-K-I silent. So skin, liver and the kidney is the correct choice, right? Identify the proteoglycan. Tricky question if you are really not interested in the figures. Chondroitin sulfate, dermatan sulfate. They both are isomers. I have told you they may have sulfate at these two positions. But they are composed of, there is glucuronic acid, right, and there is iduronate. Do you see this COOH is in like the opposite? So that's why I was telling you they are the isomers for each other, right? And in this, what they are giving you the sulfate at the 4th position. So it is what? And that is up. Who has up? Dermatan has up. I'm sorry, dermatan sulfate has COOH as the up, not the chondroitin sulfate has COOH as the up in the upward position, not in the downward position. So this is the chondroitin sulfate, not the dermatan sulfate. The COOH is down and we want the COOH that is up the plane, right? The other choices are, one is the keratin sulfate. In the keratin sulfate, you must see the galactose here. So this should be a galactose ring. That is a keratin sulfate, right? And heparin we know is the polysaccharide unit, right? And what it has, this polysaccharide has this kind of thing. Glucosamine, glucuronic acid, then glucosamine, then glucuronic acid, then glucosamine. This is repetitive in this fashion. And then there is iduronate acid, then again the glucosamine, glucuronic acid, glucosamine, glucuronic acid, then finally it will be having a diamond shaped iduronate acid. This will be the sequence of the heparin. Enzyme present in both glycolysis and the gluconeogenesis, which is common to both glycolysis and gluconeogenesis. There are many enzymes which are common to glycolysis and the gluconeogenesis. And remember this from the class, right? Phosphofructokinase is there in the glycolysis, but in the gluconeogenesis you have fructose 1,6-bisphosphatase. So this is not the choice. PEP-CK is there in the gluconeogenesis, but instead you have pyruvate kinase. So this is not the choice. Phosphoglycerate kinase. Where is the phosphoglycerate kinase? Here. It is present in both of them. Pyruvate kinase is present in the glycolysis. It is not there in the gluconeogenesis. So this is not the choice. The correct choice is C, that is phosphoglycerate kinase. If you feel it is wrong, if this table is wrong, you can just ping me. Glucose is stored in glycogen form. Why? Very tricky question, and I loved this question, though it seems very simple. Glucose is stored in the glycogen form. Why? Is it compact form? Yes, it is compact form. It can be reduced from multiple branches. Can it be reduced from multiple branches? A is the correct choice. Why B is not the choice? Glycogen is being branched, right? Many branches are there, but all the branches, they end with the non-reducing sugar residue. Read the last line. All the branches, they have just one reducing end. They do not have the multiple reducing end. So that makes this statement incorrect. It can be stored at multiple sites, majorly in liver and the muscles, not everywhere. So this is not the correct choice. Can provide glucose as much as needed when needed for 1 week? No, it is therefore 16 hours. The liver glycogen survives up to 16 hours. After that, we shift to the fatty acid oxidation. DNA methylation does not occur in, is not involved in. Again, the same kind of a question, but again, the things are changed, right? So first choice is the mismatch repair. In E. coli, the methyl-directed mismatched repair exists, right? So it is involved in there. DNA replication, does it involve in DNA replication? It is there for the gene silencing, it is there for the gene capping, it is there for the mismatch repair. It is never involved in the DNA replication. Antibody testing for CoV2 depends upon what? Antibody testing for CoV2 depends upon the, this is from the CDC government. A positive antibody test indicates what? The person has the past infection. It has the past infection, or it has been vaccinated. So that is why it has antibodies available, right? It is not used as a diagnostic test for the current infection. So these are few key features which we should be knowing for the antibody testing. So make sure you take a note for these, right? Because this is still a hot topic and we are expecting a fourth wave also. So this is the past antibody testing of CoV2 depends upon the past infection. Asymptomatic, they may give you, they may not give you, but past infection is for sure. Ammonia is toxified in the brain by converting to which of the following compounds? Okay, so there are the choices. Some problem is there. Let's go. See, if you have the ammonia in the brain, right, it is toxified by using lots of alpha ketoglutarate, right? Alpha ketoglutarate can accept ammonium ion, gets converted into glutamate. Glutamate still can have one more ammonium ion and it is eliminated in the form of the glutamine. But this leads to the decreased availability of the alpha ketoglutarate. So we have to synthesize the alpha ketoglutarate in the body, right? And that is with the help of pyruvate carboxylase enzyme and the glutamate dehydrogenase enzyme. So glutamate dehydrogenase enzyme is helping and the pyruvate carboxylase enzyme. Why we name that pyruvate carboxylase enzyme there? Because pyruvate carboxylase enzyme is doing, it is helping us to convert pyruvate into oxaloacetate, right? And then we can synthesize more of the alpha ketoglutarate, and this alpha ketoglutarate can be used for the brain detoxification. But which is involved? Which amino acid is involved? Glutamine is involved. They may give you alpha ketoglutarate, they may give you the glutamate, but ultimately it is released as a glutamine and hence it is detoxified. This is the quote for today's, and with this I'll finish this session up. To err is human. That is the first place we are in. To err is a human. गलती करना इंसान का स्वभाव है. So in the exams, in your day-to-day life, you can make mistakes. Even I can make mistakes. So mistake is a human nature, but we learn from mistakes, right? And but when you will be more mature, when you will be like when you will be guarding or maybe giving training to your juniors, then remember this point. To forgive somebody else's error is divine, right? So that will make you grow in your life. So you are going to make mistakes, lots of mistakes in the exams, in the classes. So but just learn from your mistakes and don't try not to repeat that mistakes. That's how we grow in our life, and also that is how we score in our exams. With this, I would like to thank you for your patience and welcome to all of you, and good luck and be safe from the wave. Good luck. Bye-bye.